Hey guys, this is Jason. Today we're going to be talking about olfaction, which is basically how we perceive scent and smell. And I know that based on this diagram, there's a lot going on. It looks very convoluted, but it's actually more simple than you think. If you guys would just bear with me. So I want to talk about two regions significant in olfaction, which is the olfactory epithelium and the olfactory bulb. And the olfactory epithelium is important because that's where everything's kind of being started. That's where the odor molecules first being absorbed in something called olfactory cilia, which are these hair-like structures, and they're basically just the dendrites of the olfactory neurons. So like any neuron in action potential, there's going to be action potential traveling from the dendrites of the olfactory neurons, traveling up to the axons, and then from the axons, it's going to reach globular synapses, which are basically circular synapses. And bear with me, these are called glomerulus, and the plural version of these here are glomeruli. And something significant to note is that these axons of the olfactory neurons, of the receptors, they actually converge into one glomeruli based on the neuron and the receptor. So once it reaches the synapse, then it's gonna touch the dendrites of metrial cells and the olfactory bulb here. And what that really does is that the metrial cells basically just bring the information to the brain, to the higher regions part of the brain where information will be processed. And that things that I'll be discussing later, such as combinatory coding and label line, it's going to be, the location of that is going to depend on the ecological purpose. If I'm able to just grab your attention on this particular diagram, right here, that's just basically the olfaction signal transduction pathway, and it's basically GPCR, so there's nothing brand new. So with GPCR, obviously the odor molecule, it reaches the receptor protein, and that's where the G protein is stimulated. So as you can see, if you kind of zoom in here, that's where the GDP comes out, GTP comes in, and then that triggers the activation of adenocyclase, as you can see right here. I know it's very small. And adenocyclase is an enzyme that speeds up the conversion from ATP to CCAM. And as you can see, CCAM is right here. And CCAM is a secondary messenger that prompts the activation of CNG channels, which, as we all see, basically opens the calcium and sodium channels, which allows the calcium and sodium ions to start flowing in. And obviously, once calcium and sodium flow in, that's what's causing the cell to be depolarized, which is basically creating the action potential. And so to reiterate, the CCAM binds to the action channels, right, in the membrane of the cilia, and that causes the action potential. And once the action potential is created, that's where it's really traveling from down the axon of the olfactory neurons here to the olfactory bulb, to the synapses, to the metrial cell, where it will be processed in the higher region of the brain. So that's just basically a quick run through of the olfaction signal transduction pathway. Also, just how the olfactory signal transduction pathway works. Similarly, that's also, to undo it, that's also being regulated by calcium and CAMK2, which we refer to as recovery. So 3 ways it can really recover is that the calcium or /commodulin, it binds directly to the CNG channels and it blocks them. So obviously, once you block the CNG channels, you're not going to allow the calcium and sodium influx, and it's not going to cause depolarization. So that's one way for recovery. Second way you can also undergo recovery is where the calcium commodulin, it can actually bind to PDE1C, and as a result, that convert CCAMP. Before we talked how it was conversion of ATP to CCAMP and CCAMP affinities, but now it's actually converting it back from CCAMP back to AMP, which obviously is kind of undoing that process of the CNG channels being open. So that's the second way. And then the third final way is calcium and calmodulin. They can actually activate CAMK2. That, as you can see, CAMK2 is right here. And CAMK2 can inhibit adenocyclase. And as a result of inhibition here, it downregulates the production of CCAMP, which we're seeing as a common trend now. Basically CCAMP being reversed or CCAMP being decreased, levels being going down, and as a result of that, all that is doing is preventing it, from the CNG channels to be open and an influx of sodium and calcium, which causes depolarization. So that's basically recovery, inhibition of CNG channels, and adaptation, PDIC activation. As mentioned earlier, depending on what I discussed, the higher regions and ecological purpose, I'm going to talk more in depth about that. So there's actually two olfactory systems tuned for different ecological purposes. The first one is called combinatorial system, and the second is label line system. And just to talk about combinatorial system, it's basically how we process scent. The structural elements of the odorant molecules, it can fit with different type of olfactory receptors. So there isn't really a picture of that here, but just picture a Tetris block, the L Tetris block, and picture 3 different olfactory receptors. And because hence combinatory is basically how I'm talking about, it's broadly tuned, it's not going to be an exact fit. This L Tetris block, in terms of how it fits with the different receptors, it can fit going like this, going in, which fitting into one same receptor. It can fit with this going in to another olfactory receptor. It can even fit like this going in, and you guys get the point. So the significance of the combinatorial system, because it's broadly tuned, and because it can fit, like I said, with the picture the Tetris block, fit different receptors. That's also why. What does this? So what does this mean for odor discrimination, right? So that's why we can't distinguish many smells at once. That's also why singular odors, they're not detectable in mixtures. And if we have a mixture of odors, we can't really distinguish a mix of odors from single odors because of combinatory coding, which is it's going to be random. It's going to be also known as stochastic. So now there's another system called label line system, and actually this is going to be more specific because there's going to retain more chemical specificity because it's whereas the combinatory coding was broadly tuned, there's going to be a more specific fit, and it's going to follow actually a slightly different transduction pathway. So this is going to be referring to more combinatory coding, but with label line systems, actually it involves something called, bear with me, vomeronasal organ, and that's actually separate from the olfactory epithelium. And here we can see it uses the label line system, and each receptor, as I said, it responds to one chemical. And also, as you can see here, the signal transduction is different. Instead of how they're starting with GPCR, CCAM, all that, it's actually going to be with the vomeronasal organ and then DAG, and that's where the receptors are also a little different, because whereas the combinatory coding had the olfactory bulb, the information in the transduction pathway are actually going to reach something, it's going to reach an accessory bulb. And as a result of that, that's what we call more stereotyped, because they're stronger, because they're more innate. Whereas before we refer to random or stochastic, this is going to be more specific. As you can see here, stereotype representation corresponds to innate behavior such as pheromones, and that's what this really is. I want to conclude pheromones. So pheromones, they do involve V1R and V2R receptors, and there's about hundreds of them. And the significance of that, for let's say like animals, or they respond to MUPs, which are major urinary products, and as a result of pheromones, that's where like the animals, they're able to detect certain like species recognition, male recognition, even detect sexual stage, detect whether the species is your own or not. That comes from the pheromones. And so this kind of begs the question, is there a pheromonal system in humans? So based on what I said, based on just the V1R and the V2R receptors, it's right now the science is supporting that there probably isn't a pheromonal system in humans, just based on the fact that only 5 V1R2 are intact, and all the V2R receptors, they're actually pseudogenes, and pseudogenes, they basically encode for an unknown function, or something that might not be of a function. So that's probably supporting why there isn't a pheromonal system in humans. But a counter argument is, maybe there is, because it's referring to things like menstrual cycle, and that menstrual cycle synchronization, and also in terms of how siblings they can recognize each other, even like twins, they able to detect and smell each other. So that's kind of another counter argument to what there is a pheromonal system in humans. And then another point, you may be wondering what this information is: olfactory receptors and insect olfaction. And just really briefly, it's quite simple. So for olfactory receptors in mammalians, and for example humans, we evolved our transduction involves, as I discussed, GPCR, G protein coupled receptors, and involves 7 transmembrane domains similar to visual pigment, and there's about thousands of these variations. Whereas for insects, Drosophila, the insect olfactory receptors, it's ionotropic, and it's, in terms of the receptor, it's actually an inverted topology, and this involves OR83, OR83/6 and ORX complex, which this forms the ion channel for the transduction to begin. So that's the main distinction between the both GPCR and metatropic and ionotropic. I did want to discuss that I didn't touch base on earlier in the basic rundown of this was actually, if you zoom in here, between the glomeruli, this right here is actually called the Periglomerulus, and this is involved in lateral inhibition. And well, what is lateral inhibition? Lateral inhibition is basically the neuron's response to a stimulus. It's inhibited by the excitation of a neighboring neuron. I don't know why I can't talk, but the result of that, what's the significance of lateral inhibition? So with that, because this is it's like almost like regulating the response. With that, you can have a sharpening response. So like a more discriminatory power, that's only suitable for odors that activate close by glomeruli. Another way, you can gain more control. So you're keeping cells like in the best range. And what that means is that at the same time, you're also kind of ignoring or reducing the unimportant odors. So once again, talking about discriminatory power. And lateral inhibition can also involve the timing of the meteor cells' activities, which we'll discuss here, that bring the information to the brain. And as a result of that, under that grouping, that's where identity can be defined by the combinatory code and also the timing of the activity. And that is also, the timing of the activity is known as the temporal code. Yeah, so as a result of that, with respect to identity versus intensity, so because of how we process scent in conjunction of combinatorial coding and lateral inhibition, the concentration of a certain molecule or odorant, that's also going to affect the identity of it as well. So many chemicals, they can smell very different at different concentrations. So that's what's really with respect to identity versus intensity.
Hey everybody, I'm Anya, and we're going to talk about muscle contraction because it's pretty important to motor control. So there's an interesting process that goes into muscle contraction. Essentially, sarcomeres, units of motor protein, are made up of two types of filaments. You have actin and myosin. And myosin might sound familiar. There's, like walking proteins that also have to do with myosin. So basically these, in a relaxed state, these two filaments are resting next to each other in the muscle fiber. And when an impulse occurs, calcium flows into the cell. Looking back to NMJ's neuromuscular junctions, calcium's like a big deal with signaling. So acetylcholine will lead to that calcium signaling, calcium flooding into the cell, and when this happens, filaments will slide past each other to create that contraction. So this sarcomere, this length will become this length. These outside active actin filaments will slide inwards and make it a lot shorter, so that's that sort of clenching of the muscle. Sorry to say clenching. So looking up close with this actin, there's actually several proteins involved in the actin and myosin sort of, like complexes. So there's troponin and tropomyosin, and these kind of bind and loop around the actin, and in a resting state with no contraction, they're essentially just at rest, so they're loose. And they're blocking these binding sites along the actin for myosin to connect. When calcium arrives in the cell, calcium will change the binding sites of the actin and leave this area open. So now there's room for myosin to bind to this actin. So looking at the myosin, one important feature of myosin fibers are these myosin heads, and they contain an inactive ADP and a phosphate. So in the resting state, there's a phosphate, but it's not actually contributing its energy to any process because it can't bind to anything. And as you probably know from a lot of different pathways for cascades, ATP is the energy and phosphates are the sort of energy exchange of ATP. So when this whole contraction occurs and calcium exposes those binding sites and allows myosin to be activated, the phosphate will leave, and ADP connects to the myosin sites, and the actin and the myosin are finally connected. So when myosin binds, the myosin heads will sort of change their conformation. They'll start to move, and this is sort of the drive and the energy that because of the stored phosphate energy, these myosin heads can inch their way along, and this sort of pushes the actin in, and this is what the contraction actually is. This is when the muscle is contracted. So these actin filaments here will be here. They will have moved because the myosin is inching them along. So they glide past each other in this gliding movement. After that, so what happens after this? The muscle's contracted, and wouldn't be great to constantly have contracted muscles, so we need some way to reverse the process. And this is done when ATP arrives. So this is a fresh ATP molecule with an active phosphate group, and this basically binds to the head of the myosin unit and severs the bond with that energy, and then the gliding motion is stopped. So this basically allows the myosin to sort of disconnect. The actin will probably go back to its original conformation. It'll snap back to where it was. Once this happens, ATP is at the myosin head, and it sort of dissociates into ADP and phosphate again, like at the beginning of this whole process. So the fiber is basically where it's at the beginning. So it's back to a resting state. And if calcium is still present, this just can happen all over again, depending on like the intensity of the stimulus. If you remember, other parts of firing involve intensity of stimulus to the muscle fibers. So this is something that you can imagine would be pretty relevant. And then basically everything's reset, and we can go back to the beginning and have another contraction.